Alright, welcome to the second paper. This paper is entitled The Sublethal Effects of Oil-Contaminated Sediments to Early Life Stages of the Eastern Oyster, or Crassostrea virginica. This is by Boulais in 2018 in the Journal of Environmental Pollution. Just as with the last paper, thought I might go over some definitions, abbreviations you might come across, which you might not be familiar with. Some will be familiar, such as DWH is the Deepwater Horizon. GOM would be the Gulf of Mexico. The pH is a polycyclic aromatic hydrocarbon, which are organic substances made up of carbon and hydrogen atoms grouped into at least two condensed or fused aromatic ring structures. And those are the items that we're going to be looking at today. The TPH50 actually means the total sum of 50 individual polycyclic aromatic hydrocarbons, and so that's what they're looking at there. The elutriates are simply to purify or separate out, and we'll be dealing with the oil sediments and the elutriates from that, so we'll be separating those out. The supernatant, which is simply the liquid lying above a solid residue after the processes such as centrifugation or precipitation or some sort of crystallization. The bioassays, which you know is a measurement of a concentration or potency of a substance by its effect on living cells or tissues. FSW comes across frequently as filtered seawater, not fresh seawater, but it's filtered seawater. EC50H is simply the half maximal effective concentration. So it refers to a concentration of a toxin which induces a response halfway between the baseline and the maximum after a specific exposure time. And the exposure time would be the H you see there. HEWAF, which is a high energy water accommodated fraction, are mediums containing only the fraction of petroleum that remains in aqueous phase after a mixing energy has been removed and after a predetermined sufficient time for phase separation. Again, just a few highlights of the Deepwater Horizon spill in case you are watching this one first before the second one, before this one. So again, it happened in April 20, 2010. The Deepwater Horizon Macondo oil well drilling platform exploded. 11 workers were killed. 87 days later, on 15 July 2010, the well was finally capped. It was an estimated 2.5 million gallons per day, or 240 million gallons released into the environment uncontrolled. And it wasn't until 5 years later, in July 2015, an agreement was reached between BP and the U.S. federal government and 5 Gulf states for $18.7 billion in fines, with monies to be made available over the next 15 years. So this was in the last paper as well, but simply a graphic kind of being able to put the Deepwater Horizon spill in context with other spills that are either both deliberate or accidental release. The Deepwater Horizon is the yellow and red, and then it has its associated 1.8 million gallons of dispersant as well that was tied to that spill. And then more details on each spill can be found in the notes below. Thank you. Okay, so here we have the eastern oyster, the Crassostrea virginica. Oysters are hermaphroditic bivalve that spawn when temperatures fluctuate drastically. In the wild, this typically happens during the summer, and depending on the oyster's environment or life stage, of which there are many, the oyster can be male or female, but never both at the same time. Once the oysters spawn, eggs and sperm are released into the water to be fertilized, which is what you're seeing here in the upper right-hand picture. The adult females can release as many as 5 to 8 million eggs at one time. And in the bottom right picture is the spat, and then you can see the life cycle in the diagram to the left, and you can see that there are many life stages in which a study like this potentially could target in to see, which is exactly what they do, to see how the oil sediments affect these different life stages. So here we get at the root of what is the question. Well, what are the sublethal effects of unfiltered sediment elutriates contaminated with the Deepwater Horizon oil on the fertilization success, early life stage development, and growth in the eastern oyster? And they also want to know what is the most sensitive life stages and endpoints for the ecotoxicological assessment of contaminated sediment on the eastern oyster. So why is this the question? Well, the effects of the deepwater horizon oil sediments have not been investigated in any bivalve species as of yet. The ecological and economical significance of the species make it advantageous to study and to kind of know what we potentially could lose in the event of a accidental release. The sensitivity to pollutants of the eastern oyster make it a suitable model species to investigate the effects of deepwater horizon oil sediment on the early stages of bivalve species. So why this species, the eastern oyster Crassostrea virginica? Well, the eastern oyster has not yet been evaluated for the deepwater horizon oil sediments. Oysters are filter feeders and move water and sediments through their system, which may be harmful if those particular water and sediments are contaminated. They are sensitive to environmental pollutants. And there are various life stages which allow for incremental assessments of these sublethal effects. So here we have the basic roadmap of this investigation of what we're going to be doing. So you have the eastern oyster, then you have the beaker filled with the sediments, and this is how we're going to pull the elutriates off of from those sediments. And there's a specific process in which they do that. And of course they get the individual gametes, the embryos and larvae at the different stages, and those are all examined to the elutriates, and then they're analyzed for the total pH for toxicity and effective concentrations, which we'll discuss here in the later slides. So here we have some sediment characteristics and some of the analytical chemistry of the elutriates. They were collected in 2012 from a reference site called the Loomis 2 in Louisiana. The contaminated sediments were collected from a site called the Black Hole in Louisiana, an area classified during the NRDA as having a heavy, persistent oiling. And after field collection and before toxicity testing, subsamples were sent to labs for analysis on the pH, alkaline pH homologs, and related hetero compounds using gas chromatography with a low-resolution mass spectrometry with selective ion monitoring. That's the GCMS-SIM, based on the U.S. Environmental EPA method 8270D. Subsamples for metals using EPA method 6010C for metals and 6020A for antimony and silver, and then 7471B for mercury, and then the 808IB for pesticides, the 8082A for polychlorinated biphenyls, which you know as PCBs. And they also analyzed for total organic carbon, and then particle size and total solids were also analyzed. Unfiltered water samples at various concentrations were used for toxicity testing to include control samples. And then temperature, dissolved oxygen, salinity, ammonia, and pH of elutriates were measured daily using an optic probe, a refractometer, and a pinpoint pH monitor. So the preparation of the unfiltered sediment elutriates, the samples were UV sterilized in glass beakers. Then you had a 0.1 micrometer filtered seawater, which is a FSW, filtered seawater, adjusted to 25 degrees Celsius and 22 PSU, or practical salinity units, which is a unit based on the property of the seawater conductivity, is equivalent to the per thousand. They were added to the control or contaminated sediment in a ratio of 10:1. Then it was mechanically stirred for 300 rotations per minute for 6 hours, after which there was a 12-hour settling out period for the sediment. The supernatant elutriates were siphoned off and mixed with filtered seawater in dilution series to nominal concentrations of 100, which is no dilution of stock, and 50, 25, 12.5, 6.25, 3.125, and 1.5625% of supernatant. The control elutrient was prepared using the same methodology above, except that the supernatant was not diluted. And an additional filtered seawater control with no sediment was also tested. Solutions of sediment were not filtered or centrifuged for the entire test. Collection of oyster gametes. The oyster Crassostrea virginica was collected in September from Estero Bay, Florida. The average weight was about 75 grams, and they were kept in the ambient salinity seawater for about 2 weeks. Seawater was sand filtered. The animals were fed cultured microalgae of 3 different species at a daily ration of about 3% of the oyster dry body weight. Oyster ripeness was determined by a microscopic observation of the gonadal smears, and the oocytes and the spermatozoa were examined for motility, shape, and absence of the estrias oocytes. The gametes collected by scraping gonads with a scalpel into 50 milliliters of filtered seawater, and then the sperm was sieved through a 50 micron meter mesh from 3 males and pulled into 500 milliliters of filtered seawater. And the oocytes were collected from 3 females, sieved and pulled into 2 liters of filtered seawater. And then those gamete concentrations were determined by microscopic count via the Sedgwick Rafter, a 3 by 100 microliter stage. So we have a sediment elutrient exposure of the early life stages. Here we have the gametes, embryos, and larvae were exposed to the different concentrations of the deep water horizon elutriates, the control elutriates, which was with fresh sediment, clean sediment, and then the filtered seawater control, which had no sediment at all. There were 4 replicates of each treatment exposure at the standard 25, give or take, a degree Celsius, and at 21.5 practical salinity units. The elutrients were not renewed, and no aeration was provided during the experiment except for the veliger larvae, and which they were exposed for 48-hour exposure, and so they received a gentle aeration of about 60 bubbles per minute to maintain DO, which is dissolved oxygen, above 4 milligrams per liter. So for the statistical analysis, the results are presented as a mean standard deviation, log logistic models with the DRC or the dose response curves packaged in R version 3.1.1 at 2014 to fit dose response curves. A 3-parameter log logistic model was fitted for binomial response variables, those being fertilization and abnormality. A 4-parameter log logistical model was fitted for shell length. The estimated concentration from fitted models for relevant quantiles, and all results are reported with a 95% confidence interval based on profile likelihood using the BBMLE, which is the Ben-Bolker maximum likelihood estimation. So what did they find? Well, the chemical and physical characteristics of the field collected sediments are listed here in Table 1. The filtered seawater used for the control contained very low levels of PAH, which is the polycyclic aromatic hydrocarbons, at background levels, where the total for all 50 individual pH was at 0.08 micrograms per liter, give or take 0.03. Note that the fines, which is clay plus silt, of the contaminated sediment could not be assessed because of the high oil content. So you won't. So that's why you see a Na under the fines for the contaminated sediment. So here we have the composition of PAH in the unfiltered sediment elutrient was very similar to the composition of pHs in the field collected deepwater horizon contaminated sediment. This indicates that the pHs in the unfiltered sediment elutrients were likely from the pHs on the suspended fine-grained particles rather than the pHs from the water accommodated fractions, which is a medium containing only the fraction of petroleum that remains in aqueous phase after mixing of energy has been removed, and after a period sufficient for phase separation. So that's what that means. So it means if you look on the scale here, you can see that each of these constituents, like N1, N2, N3, and 4, are very similar. So they track the black filled-in bars are the contaminated sediment, and then the unfilled-in bars are the unfiltered sediment elutriates. And so you see how they track very closely to its pair constituent. So here we're going to look at sublethal effects on fertilization. You notice on the graph here on the left, you have a 20 and a 50. So those are the effective concentration numbers that you're looking at. The gray filled circles outlined in red are sediment and nutrient control, and the black filled circles outlined in green represent seawater control. This will be the same theme throughout these next 3 or 4 slides in the graph. You'll also notice that the horizontal lines in red correspond to the 20 and the 50 on the left. So I added those just to make it easier to see. So you have filtered success on gametes exposed to filtered seawater. It was 90%, and the control sediments were about 87%. At the highest dose of unfiltered sediment, the elutriate tested at 152.8 micrograms. And there was about 54% unfertilized. And then you have fertilization decreased with an increasing dose. So where you had the effective concentration after 1 hour was 40.6, and then the effective concentration 50 after 1 hour was 173 micrograms. So here we have the sublethal effects on embryogenesis and early larval development. This is figure 3 broken up into 3 different squares to make it a little more manageable. Fig 3A, which is the upper left, is gametes, and they were exposed for 24 hours. 3B, the upper right, is embryos, and they were exposed for 24 hours. And in figure 3C, the bottom right, were the veliger larval, and they were exposed for 48 hours. And just like in the last, the gray filled circles outlined in red are sediment and nutrient controls. And then the black filled circles outlined in green represent seawater control. The dose response curves were fitted using seawater control. The horizontal lines on the curves represent the 95% confidence interval of EC20, or the effective concentration, and effective concentration 50. A high percentage of abnormality and larval development from exposed gametes to sediment and nutrient control did not allow for determining the EC20 and EC50 values, so that's why you don't see those. The low percentage of abnormality at the highest doses of sediment and nutrient tested in the veliger exposure did not allow for calculation of the effective concentration 50 value. And if you look at table 2, you see the EC20 for the embryo was 77.7, and then for the veliger was 95.9, and then EC50, or effective concentration of 50% inhibition, was 151 for the embryo, and it was not able to be obtained for the veliger larval stage. So here we have sublethal effects of larval size. This is a figure 4A broken into 3 with the same sort of pattern as we did in the past, where figure 4A is gametes for 24 hours, figure 4B is embryos for 24 hours, and 4C, the veliger larvae, were 48-hour exposure. Now you'll notice that there's an absence of data at the highest concentrations of unfiltered sediment and nutrient was due to the high mortality. So the second highest concentration tested was 72 micrograms of total PAH at 50, with the mean length of 59.8 micrometers. In the 48-hour exposure of veliger larvae, sediment-associated PAHs reduced the larval shell to about 70.7 micrometers compared to controls, which measured in at 75.5 for the filtered seawater and 73.8 for sediment controlled, respectively. So the unfiltered sediment and nutrient exposure induced a dose response decrease in shell length for larval development from exposed embryos, with an EC20 after 24 hours value of 1180 micrograms of total pH. So some sublethal effects of sediment-associated pH on early life stages. Fertilization was reduced after exposure of gametes to unfiltered sediment and nutrients. The sediment-derived pH induced abnormal development and reduced shell length and 24-hour-old larval development from the exposed embryos. This study found that the control sediment nutrient induced abnormalities in 24-hour-old larvae developed from exposed gametes. The chemical analysis found that no metals, pesticides, PCBs, and the TPH50 in the sediment nutrient control, which is the clean sediment, but there was high percentage of fine particles of silt and clay, about 82%. The fine particles in the sediment nutrient control may have impeded early embryogenesis after the first hour post-fertilization, without impacting fertilization success and later embryogenesis. The results of this study suggest that exposure to fine particles during the first hour of embryogenesis may cause abnormalities in the eastern oyster. The mechanism is unknown, but suspect particles bind to the membranes and interrupt cell cleavage. Toxicity of sediment-associated pH is compared to the high-energy water-accommodated fraction-derived pH on early life stages. The material and methods used in this present study were the same as those used in Vignier et al. In 2015, allowing the comparison of effective concentrations for inhibition on fertilization ability, embryogenesis, and larval development of the eastern oyster between high-energy water-accommodated fraction and unfiltered sediment nutrient preparations from the same deepwater horizon oil. The abnormality induction and larval development from exposed embryos came in at the effective concentration of 20% after 24 hours was 218 micrograms, and the effective concentration 50 after 24 hours came in at 342 micrograms for the high-energy water-accommodated fraction. The increase in toxicity in contaminated sediment nutrient cannot be explained by the fine particles in the water column alone, because the same fertilization success was seen in the control treatments. So the pHs attached to the fine-grained particles is suspected. There was found a twofold higher percentage increase of three-ring pHs, i.e. DBT4, and four-ring pHs, the MBT1, MBT2, and MBT3, which are just the alphanumeric acronyms for those compounds. So they were suspected to be to have low water solubility and high affinity for organic carbon, limiting the degradation of those particular pHs. The toxicity of individual pHs on oyster early life stages remains unknown, and further research is needed to tease out the individual pHs that may cause these toxicity. Sensitive life stages for ecotoxicological assessment. 1 hour fertilization success and 24-hour abnormality of larval exposed as embryo endpoints are more sensitive than shell length across all bioassays. It's also found that both fertilization success and embryogenesis endpoints showed a strong dose-dependent response for the elutriates of oil-contaminated sediment, indicating both are sensitive sublethal endpoints for assessing acute toxicity of oiled sediments. Fertilization success and abnormality of larval exposed as embryos were the most sensitive endpoints for assessing the toxicity of oil-contaminated sediment. So in conclusion, this study presented the first investigation for the effect of unfiltered elutriates from sediment contaminated with deepwater horizon oil on the early life stages of marine bivalve species, specifically the eastern oyster, Crassostrea virginica. Gametes, embryos, and villager larvae of the eastern oyster can be adversely impacted by the oil attached to sediments. Fertilization success and 24-hour abnormality of larvae exposed as endpoints were the most sensitive endpoints for ecotoxicological assessment of oil-contaminated sediment to early life stages of this species. Here are the references that I used, with the top one being the actual paper itself that was used for this presentation, followed by several web links to either information and or images that were used throughout this presentation. I hope you enjoyed the presentation and found it very interesting. I know I did, and I learned a lot about some of the mythologies and some of the things that I actually do where I work. I do a lot of metal testing at the steel mill where I work at, and so it was interesting to come across some of these same EPA method tests that were done in this study as well as what I do at work as well. So it was very interesting to see that. Again, thanks for tuning in, and good luck. Thank you.